Hello, everyone. Thank you very much for joining us today at this Biomedicines webinar. Today, we are going to talk about antimicrobial peptides, aka host defense peptides. And we have three guests, the chair, Dr. Jitka Petrlova, and two speakers, Dr. Parvin Paparedi and Professor Dr. Esther Bois. I will talk next a bit about MDPI. MDPI is pioneering scholarly open access publishing and is supporting academic communities since 1996. It is based in Basel, Switzerland, and has a mission to foster open scientific exchange in all forms across all disciplines. It hosts over 400 diverse journals, which are all open access. 413 are peer-reviewed and 9 are conferences journals. All these journals are supported by more than 100,000 academic experts who share our mission, values, and are committed to provide high-quality service for our authors. Our journal, Biomedicines, is an international scientific and peer-reviewed open access journals focused on biomedicines and is published monthly in MDPI. In this table, you can see various factors such as the impact factor, the site score, and in days, the time to first decision and acceptance to publication. For today, this is the schedule of the webinar. Dr. Jitka Petrlova will have a short introduction of the topic and of the speakers. Then Dr. Parvin Paparedi will have his presentation about the coagulation system-derived antimicrobial peptides in host defense. Afterwards, Professor Dr. Esther Bois will talk about moonlight RNases, a source of antimicrobial peptides. After both talks, we are going to have the Q&A session. And at the end, Dr. Petrlova will close the webinar. I would like to kindly ask you to post any question you might have for the speakers in the Q&A box. And to please remember to write to whom you are addressing the question to. The relevant special issue related to the webinar is the special issue antimicrobial peptides, aka host defense peptides, from basic research to therapy. And this is the second volume of the special issue. You can find both the volumes on our website and read the published articles. Dr. Jitka Petrlova, Associate Professor. After completing her PhD in biochemistry at the Mendel University in the Czech Republic, she has been a postdoctoral researcher at the School of Medicine at the University of California, Davis in USA. She joined the medical faculty of the Lund University in Sweden in 2010. And she was always fascinated by the aggregation of protein and peptides and the links to diverse physiological and pathological processes. Her current research is focused on the aggregation as a novel host defense mechanism of relevance for skin inflammation and wound healing. In particular, she studies host defense roles of blood proteins belonging to the apolipoprotein group, such as apolipoprotein E. And her findings demonstrate a previously undisclosed role of apolipoprotein E involving aggregation of endotoxins and bacteria, enabling clearance of bacterial toxins and microbial killing. The link between host defense and aggregation also suggests that persisting inflammation may lead to dysfunctional activation of such protein-based host defense pathways. And therefore, her research also raises interesting perspectives on the relationship between antimicrobial and amyloidogenic peptides. Next, I will kindly ask Dr. Petrlova to do an introduction of the topic and of the speaker. Dr. Petrlova, you have the microphone, so to say.
I would like to welcome everyone as well to this webinar on antimicrobial peptide and host defense peptides. And it's my great pleasure to introduce you to you all our two speakers. We have Associate Professor Praveen Paparade, who is also a dear colleague of mine, and he is working at the Division of Infectious Medicine at the Department of Clinical Sciences at Lund University in Sweden. And Praveen is interested very much on studying the dysregulatory host defense mechanisms and developing a novel biomolecules-based therapies, drugs targeting bacterial infection, and in the sense of chronic and acute inflammatory diseases. He also published a lot of work on antimicrobial peptides derived from the factors of coagulation system, which he is going to talk more today. We have the second speaker, which is Professor Esther Boksic, and she is working currently at the Department of Biochemistry and Molecular Biology at the Faculty of Biosciences at the University of Barcelona in Spain. And she is an outstanding researcher, well-published researcher. Her interest is in structural and functional characterization of antimicrobial peptide and protein, and her main research focus is on the mechanism of action of human antimicrobial RNAs and potential development of novel antibiotic agents as it's needed to fight the bacterial resistance. And she's going to talk today about RNAs as a source of antimicrobial peptides. And before you will hear from both of the speakers today, I would like to guide you through a little bit of the basics of the field of antimicrobial peptides. As you may know, we are from head to toe covered by bacteria. We have on our epithelial surfaces much more bacteria than our own cells. Those bacteria we call good bacteria, commensals. And if you look at the skin, the ratio, it's actually even greater for bacteria, as the skin can stretch up to 100 square meters if we include all pores, wrinkles, and hair follicles, for example, which gives bacteria a lot of space to be able to grow on. And these commensals are important for us. They help us with digestion, vitamin productions in guts, and they are also important for space filling if they block physically the access of pathogens. And be able to keep this balanced relationship between host and bacteria, we have innate system to keep it in check. In vertebrates, as we have innate immunity, which is activated rapidly and is not specific. And then we have adaptive immunity, which is activated with delayed and is specific. Our host defense peptides or antimicrobial peptides are part of the innate immunity, which is evolutionary older system and exists on different levels in all multicellular organisms. We know that the antimicrobial peptides can have quite differences in the size and the structure. They have positive charge, which allows them strongly interact with the negatively charged surface of bacteria, which cause disruption, form formation, leakage, and death. If we look at the mammalian cells, we don't have a strong negative charge on the surface. We have different lipid composition, and the interaction is much weaker, which means that the concentration of peptides, antimicrobial peptides, which is deadly for bacteria and toxic, it's harmless for our own cells. Then today we don't talk just about antimicrobial peptides, we talk about host defense peptides. Due to their multifunctionality, they are not just antibacterial, they can be antiviral, fungi, cancer, they can be antiseptic, they can have immunomodulatory property, they can scavenge ROS radicals, and they can also scavenge neutralize endotoxins. And this multifunctionality gives them the potential to be developed for new therapeutic agents against bacterial infections. And what is obviously needed due to bacterial resistance. And I am pretty sure you will hear more about those things from our today's speakers. And this is my last slide, and I would like to ask the next speaker, Praveen, to share his presentation and show us his outstanding research.
Good morning, everyone. And first of all, I would like to thank the organizers for giving me the chance to present my work here. And also thanks Itka for a nice introduction for the antimicrobial peptides. So today I'm going to talk about coagulation system derived antimicrobial peptides in host defense. So to keep it simple, as Itka has already introduced about the antimicrobial peptides and innate immunity, so I'm just keeping as like an overall just brief introduction. So since I'm also belong to dermatology, I worked before. So I talk more about the wounding and then the surroundings where you could find the antimicrobial peptides. So the best thing is like during the innate immune system, coagulation and complement is the two arms of the innate immune system, where coagulation is activated by intrinsic pathway and extrinsic pathway. And this leads to the activation of prothrombin to thrombin, and thrombin can cleave soluble fibrinogen to fibrin, which then form clots. And whereas complement can be activated by classical alternative on lectin pathways. But when it comes to the coagulation and complement interactions, so they can also activate each other. For example, thrombin can cleave C3 and C5, and which can lead to the complement activation. So what happens during the wound healing? So when the skin barrier is broken down, and of course the blood flow to the wound, and when the wound is trying to like healing process, there will be a lot of proteins that are accumulated inside the wound, and these proteins can be degraded by the host enzymes or by the bacterial enzymes, and then there will be a lot of endogenous fragments generated during this wound healing process, and these fragments can act as a host defense peptide. So this slide, as Itka has already mentioned, one thing that I would add is there might be a small difference between the sizes and then the structure, but as she mentioned, like most of them has this positive charge and amphiphaticity. And one peptide can have a multiple functions of this host defense functions. It can be anticoagulant, and it be antifungal, or it can be antiviral, and it can also have anti-inflammatory. So today I'm going to talk about three projects that I have worked during my this inflammation, and then the host defense peptide. So one project that I'm going to discuss is thrombin-derived antimicrobial peptides that has both anti-inflammatory and antimicrobial properties. So thrombin is a central coagulation protein, and it is highly expressed in liver, and it is present in plasma. And then, of course, it converts, as I said, like fibrinogen to fibrin, and it has several other functions. When you look at the structure of the thrombin, you can see the C-terminal is highly charged and has an amphiphaticity. So the question is whether this thrombin is involved in the antimicrobial activity or the host defense functions during the wound healing. So what we have done is we raised an antibody which is specific to the C-terminus, and we checked whether if the C-terminal peptides are generated in the wound healing process. So what you could see here is This is wound fluid from acute wound fluid and chronic wound fluid from patients, and you could see several fragments are generated during the process of wound healing. And we could also repeat the same thing in vivo, where we could incubate plasma and fibrin with the neutrophil elastase, which is a high abundant enzyme that is present in wound, could also cleave the thrombin and then generate the C-terminal fragments of thrombin. So next we checked whether these C-terminal fragments are antimicrobial or not. So what we have done here is that we incubated thrombin and prothrombin with neutrophil elastase, and then at different time points. And then, as you can see in the SDS page, you can see several fragments are generated during the neutrophil elastase cleaving. And then when we incubate when we add this to an irradiated diffusion assay, you could see in the right side where you could see the zones of inhibition. So the bigger the zone means that the better the antimicrobial activity. And you could see within 5 minutes of the incubation with prothrombin or thrombin with neutrophil elastase can generate the antimicrobial zones. So we have also tested that whether the C-terminal part of the peptide can be a therapeutic application. So here what we have done is we injected the mice with Pseudomonas aeruginosa, and then we treated the mice with the peptide, where you could see that the peptide GK25 could kill the bacteria, and the mice could have a higher survival rate compared to the untreated mice. And you could also see that the bacterial CFS has reduced during the peptide treatment. And later we could also see if this peptide has anti-inflammatory activity. So what we have done here is we incubated peptide with either Pseudomonas or E. coli aeruginosa LPS, and in monocytes, and you could see the enough kappa B activity in the presence of the peptides and then the LPS alone. And you could see that, like, the peptides activity, the peptides, the dose, higher the dose, that you could see the reduction of the LPS stimulation. And when we did the in vivo experiment where we injected LPS in the mice and treated with the peptide, you could see the higher survival rate, and this could lead to the reduction of cytokine storm in these mice. So when it comes to the my second project, what we have, I'm going to present the TFPI-derived peptide, which has anticoagulant and that helps for the killing the bacteria with the help of complement activation. So what is the TFPI? It's a Kunjan type serine protease inhibitor, and of course it's present in plasma, and it is expressed by several cells. And it's, of course, it's anticoagulant activity by inhibiting the tissue factor and factor 7 complex. And then it is highly upregulated during bacterial toxins or inflammatory conditions. So if you look at the structure, it has 3 Kunjan type domains, and it has highly negatively charged N-terminus and then the highly positively charged C-terminus. And it has shown previously that several proteases like thrombin, plasmin, or bacterial enzymes can cleave the C-terminal part of TFPI. And the TFPI-1 protein exists either in full length or the C-terminal truncated form in normal conditions in vivo. So when it comes to wounding, where you could see that TFPI is highly upregulated in the endothelial cells, layer cells, and you could see that during the acute wound and chronic wound, you could see really high upregulation of TFPI. So this confirms or suggests that TFPI can play an important role in the host defense. So what we have done is here, when we synthesize the peptide and when we check the antimicrobial activity, so here you can see that in E. coli, in the normal buffer conditions, compared to the citrate plasma conditions, where you could see the peptide is highly antimicrobial in the presence of plasma compared to the buffer. And this is quite different from other coagulation, other peptides, because the peptide can interact with different proteins since they are charged, and then they lose a little bit activity. But here in this case, it's quite opposite. The plasma, in the plasma conditions, the activity is higher than in the buffer conditions. And this activity is lost when we heat inactivate the citrate plasma. So this completely, we believe that this peptide is involved in the activation of complement. So next, what we have done here in this experiment is we incubated the same experiment, but we isolated the surface bound proteins from the bacteria. And then when we detect the C1q, the classical pathway complement factor, and we could see that in the presence of peptide, you could see the higher the C1q is bound to the bacterial surface when compared to the control alone. And of course, then when you have the more C1q, then you obviously you have more the MAC complex deposition of C9. And this, we could also see that like you have the C3, when the complement is activated, you have a cleavage of C3a, and this also involves in the antimicrobial activity. And this you could see in the electron microscopy. So the next thing is like, we thought like, okay, maybe this could be a really interesting peptide to go for a therapeutic function. So we did a first round of experiments in the mice, and then the peptides completely failed in the mice. So we thought like, what happened? And then when we did this ex vivo condition or in vitro conditions, where we incubated this peptide in mouse plasma, we couldn't inhibit the we couldn't kill the bacteria, and then the complement activation is completely lost. You could see in the human plasma, you could see higher killing activity when compared to buffer alone, whereas in mouse, 2 different C57 and bulb C mice, the activity is completely lost. So we thought like, okay, what could be the reason? And then the next thing we have seen is that when we look at the sequence between the human and then this mouse, they are completely distinctly related between the human and then the mouse. So in another study, what we have done is we looked further how the role of this is playing during the evolution of the TFPI and then the host defense mechanism, this one. So if you look at in the phylogenetic tree and all vertebrates, you could see that like the TFPI is highly conserved, and whereas if you look at the C terminus, you could also see mostly 80% of the sequence of C terminus is highly conserved between all vertebrates. So then what makes them unique between the activation of complement between the species? So what we have done is we synthesized peptides from several species like birds peptides and reptiles and mammals. And before we conclude that, like, you know, C1q and IgG is important for the complement activation. What we have done is we use an IDS enzyme, which is derived from the asparaginase, where it could cleave the IgG in FAB and FC region, and it will inactivate the IgG function, and that could escape from the complement. So what we have done in this experiment is we incubated IDS in plasma, and then we tested the antimicrobial activity of the peptide, the GGL27, where you could see that, of course, if you have plasma and then, if you have in the buffer alone, you don't, the plasma alone, you have 100% of, if it's put it in survival, you have 100% survival. And then in the presence of peptide, you see a higher antimicrobial activity. And then if you have the IDS, in the presence of IDS, the peptide activities is completely lost. So this completely suggests that the peptide is involved in the complement activation, and which could aid in the antimicrobial activity. So, and then when we compare this this experiment with the evolutionary, so we synthesize the peptides, as I mentioned, like different mammalian peptides, bird peptides, reptile peptides, and fish peptides. And then we have tested their antimicrobial activity in two different conditions. One is within their host or in the presence of human plasma. So you can see here in the right side, if you have the fish peptides cannot activate or kill the bacteria in their plasma, or the reptiles or the birds. But whereas in mammals, except rodents, all peptides, mammalian peptides, could activate the complement activation within the host plasma. For example, a monkey peptide in the monkey plasma, or bovine plasma in a bovine, bovine peptide in a bovine plasma could kill the kill the bacteria. But whereas all the peptides from fish to the mammals, peptides could activate the complement activation in human plasma. So this suggests that it is correlated with the complement evolutionary also. If you look at the evolutionary of the complement, only the classical pathway is present in mammals, but the lectin pathway is present in all of them. So the classical pathway is the one that requires IgG or the immunoglobulin to get activation. And if you look at the immunoglobulins here, only the IgE and IgG is missing in the birds, reptiles, and fish. So if you look at the evolutionary of this immunoglobulins, so the IgG and then IgE is only present in mammals, as I showed before, but it's not present in all remaining species. So this, and so we know that the peptide binds to the immunoglobulins and then activates complement. How this can bind? So what we have done is a computational modeling where we could see that this is the peptide which could bind to the FC region of the IgG, and then this could change the conformational change where it could help to bind a faster and a better way of the C1q binding. And this could be one of the reasons why this peptide activates complement faster in the presence of this peptide. So, and then coming to my other last, so I'll just keep it simple. I hope it's, yeah. So the other proteins involved in the host different functions. So it's not necessarily the fragments are generated from the proteins, or could be antimicrobial, and some protein can also be like intact or activated, or different forms of proteins could also be antimicrobial, or it could also have 8 different host different functions. So I'm going to give 2 examples in this topic. One is heparin cofactor is also a serine protease inhibitor. So we have also tested like this protein also undergoes different the cleavage from the different proteins, and then this peptide generated from this protein can also be activated as an anticoagulant, but anticoagulant and antimicrobial and anti-inflammatory. But here, what you could see is also the protein, the intact protein. We could also see the isoform. Like if you have a smaller instead of smaller fragment, there is like this protein is 55 kilodaltons. If it cleaves like and have like 35 or 40 kilodaltons, protein could also act as a antimicrobial. And this is just an example of the paper that we have published, where you could see if you inject the bacteria to the knockout and wild type mice, you could see higher CFUs in the knockout mice when compared to the wild type. And of course, you could also see the higher levels of cytokines in the knockout mice. And the next one is the antithrombin. So the antithrombin is also a serine protease inhibitor, and actually it is expressed in 3 different forms from all individual from a different species, from the snakes to the humans, as far as I know, and it expresses latent, alpha, and beta form. The difference between 3 is the glycosylation. So it's just only few glycosylations. So the small change in the glycosylation can aid in the better coagulation and better antimicrobial and better anti-inflammatory. And this is what we have published. And you could see here, this is just for a proof that you could see here, this is a LPS. So if you have like a normal alpha antithrombin, and this is the beta antithrombin, you could see that like it has better antimicrobial and better inflammatory and better coagulation. And the antimicrobial, when it comes to antimicrobial, you could see that like beta antithrombin can kill the bacteria and can aid to the survival. And whereas you can also see in the live imaging IVs, so you could see that low CFE levels in the mice compared to the E. coli alone are treated with the alpha. So I just want to conclude my presentation. So as I said from the beginning, the proteolytic cleavage of proteins during the wounding, and of course the coagulation proteins can generate several antimicrobial peptides, which can act as host defense. And these peptides can be a potential therapeutic candidates. One, the 2 projects that I mentioned, thrombin peptides, are already in kind of clinical trials, preclinical trials, and it is the results are quite interesting. And there, the Exinate company is producing that. And then the antithrombin that I mentioned in the last one, the Octopharma is quite interesting to do any preclinical trials in the sepsis patients. So with this, I would like to thank everyone for your attention.
Thank you very much for your presentation, Dr. Papareddy, for introducing to us your research. As we will have the Q&A session at the end of the presentations, I would like to ask Dr. Bois if she could share with us her screen and share with us her presentation.
Well, I want to thank you for the invitation to participate in this webinar, and also Dr. Pavlova for the invitation, and the Biomedicine Journal for organizing the webinar. I think the first talk has really been very interesting for me from my side, but I will wait for the Q&A question session because I have a lot of questions for Dr. Parvin. So my work is related, as the title is indicating, on ribonucleases. So these are a group of antimicrobial proteins, and they can be also a source of antimicrobial peptides. I'm working at the Autonomous University of Barcelona, and which is a campus which is located outside Barcelona, at the other side of the mountains. So here you can see a bit the skyline of Barcelona. So antimicrobial peptide, they have a rapid and multifaceted mechanism, which is is a good property to avoid the emergence of bacterial resistance, which is one of the main health problems we are having right now. And it is considered that by 2050, it can be the principal health problem if no action is taken. So there are a lot of superbugs, multi-drug resistant bacteria that are emerging to conventional antibiotics. So we need to find out alternative strategy. And in particular, the gram-negative, and such as Acinetobacter baumannii, which is one of the bacteria that we are working. So we are working on antimicrobial protein, which are from our own immune system, so they should give provide a better effect with reduced or non-toxicity, because they are from our own innate system. So usually this antimicrobial protein and peptide, they have this multifaceted mechanism which combine a direct action at the bacterial wall together with an intracellular target and some immunomodulatory actions. So in our group, we are working on antimicrobial ribonucleases, which they are small secretory proteins. They are secreted by innate cells, which can be either epithelial or different blood cell type cells, and they are protecting body fluids. So they create like a barrier to infection. In humans, we have 8 canonical ribonucleases, so we have 8 variants. And this is a vertebrate-specific family. So it's quite interesting for evolutionary studies to analyze how these different RNases have evolved to adjust to the different properties to fight during us defense, to fight the different pathogens. So you all know the main reference family, which is bovine ribonuclease A, which was one of the principal proteins of study by biochemistry methodologies, and 4 Nobel Prize were awarded related to works on this protein. So bovine pancreatic ribonuclease was obtained from slaughterhouse, and they could purify around at the range of kilogram yield. So this was very useful for these first studies. And for example, is the protein where amphisem could correlate the primary structure to the 3-dimensional fold, and also one of the first X-ray crystallography studies, but even the kinetic mechanism, which is an acid-base catalytic mechanism, was deduced before the 3-dimensional structure. So it's a family around this reference member. But initially, it was considered that this bovine pancreatic ribonuclease would work by eliminating a huge amount of RNA, which accumulates at the digestion system, at the digestion gut. But later on, the evolutionary study realized that the origin of the family, and probably the main function of the family, was not to remove an excess of RNA that was accumulating, but was more related to host defense, as it was found, for example, in fishes and amphibia and reptiles. So as I was commenting, in human, we have these 8 ribonucleases, and interestingly, they are expressed by different cell types and under different stimulus. So the stimulus usually are indicator of a stress condition. The cell is under stress condition, and this stress stimulus can be either infection, but also like tissue injury. For example, the wound healing, wound injury, or inflammation, sepsis, different conditions which induce the selective expression of one of these ribonucleases. So we can see, for example, for virus infection, we have a specific ribonuclease that's activated, and by bacterial infection, we have a different ribonuclease which is activated. Not only they are working by eliminating the excess of RNA at the extracellular level, but they can also be internalized and through the endosomal system and they can participate in the removal of pathogens at intercellular level. So, for example, the accumulation of pathogen RNA. And interestingly, researchers have related the cleavage of RNA at the endosomal level with the activation of TLR and these immunomodulatory properties. So, on the other hand, we have to consider that the cells at the cytosol, they have an inhibitor, which is interacting with RNAs at with a very strong affinity at the picomolar, nanomolar range, and it's embracing totally, as you can see here in this on the right slide, it's embracing totally the ribonuclease. So this inhibitor is found at the cytosol, and it's protecting all the messenger RNA, different tRNAs, the RNAs that are located at the cytosol, and it's avoiding the toxicity of these ribonucleases. This is an inhibitor that is expressed at high yield in most of all, or nearly, we can say, all somatic cells. So, well, now talking more closely to the protein we are working on, which is ribonuclease 3. Ribonuclease 3 is expressed mostly in eosinophils. So it's called also the eosinophil cationic protein, and has derived from a common ancestor together with ribonuclease 2. So we have ribonuclease 2 and 3 secreted by eosinophils that has derived from a common ancestor. Ribonuclease 2 is also called the eosinophil-derived neurotoxin. So we have these two eosinophil RNases. And what is interesting is that during evolution, these two proteins has evolved at evolutionary talking very short period of time during this primate, this group of primates, and this ribonuclease 3 has acquired a very high isoelectric point. So it has gained a high number of arginines. It has 19 arginines in a sequence of only 133 amino acids. And what we can see is that during this evolution, this protein has acquired this high cationicity in detriment of the catalytic activity. So this means that this protein has a much reduced catalytic efficiency, but has acquired this bactericidal activity. Here we can see in these pictures how RNase3 has this cationic patch, which is not observed in ribonuclease 2. We still have these 8 arginines, but there is a huge difference between RNase 3 with 19 arginines and RNase 2. Not only this protein has a high number of arginines, but it has also some hydrophobic patch. Like, for example, we can see this tryptophan, which is surface exposed. So we have this high isoelectric point combined with this combination of cationic and hydrophobic patch. So we can say this amphipaticity, it's very convenient for its antimicrobial activity. If we do an analysis of conserved versus non-conserved residues, we can see that the active site residues are are very conserved. So we can see in this, in this dark red color. And on the other hand, the surface residues, cationic or hydrophobic, they are non-conserved within the family. So they are specific for this antimicrobial ribonuclease. So this RNase3, it has a high antipathogen activity, It's a wide range antipathogen activity, so it has activity against helminth, fungi, protozoa, bacteria, and virus. In our work, we have worked in the different with different targets. For example, like here I put only two brief examples in Leishmania, or for example in Mycobacteria. But today I will talk about the bactericidal action in bacteria, and in particular in gram-negative bacteria. First of all, I want to talk about the studies we did initially to correlate the amphipaticity of ribonuclease 3 to its action on model membrane. So this was the first, we were not pioneer to study the non-related, the activities non-related to the enzymatic properties of ribonuclease 3. So we could see that unrelated to the catalytic activity, we have an interaction of the protein with model membrane. We did some studies with brominated phospholipids, and we could analyze the internalization of the protein in model membrane and the importance of anionic phospholipids that could interact with these arginines. We also tested our protein in vesicles with different compositions of phospholipids, and we could see how the protein was inducing the aggregation of the vesicles. This cationicidity of the protein was also useful for its interaction with the molecules that are exposed at the bacteria wall level. So for example, in particular in gram-negative, we have the lipopolysaccharides, and there is a very high binding affinity, which I will show later, some results. We have a high affinity to lipopolysaccharides, and we can see also by electron microscopy how the protein is inducing the lysis of the bacteria wall. We also studied the induction of aggregation of bacteria, of gram-negative bacteria, induced by the binding of the protein to the bacteria. The aggregation is leading bacteria death. So first of all, I also would like to talk about an aggregation patch that we identify in this protein. This aggregation patch was identified by using the AgreScan server, which is an algorithm that was developed by researchers, Salvador Ventura, from our university. So they have this online server. And we can see how at the N-terminus of our protein, there is a patch specific for ribonuclease 3, which is not found in ribonuclease 2. This patch, as we can see here on the right, it's in this helix at the N-terminus, and we could identify a single residue, isoleucine 13, which by substituting this residue by an alanine, we can switch off the aggregation patch. This aggregation patch is involved in protein-protein interaction and is inducing not only the protein aggregation, but also the agglutination of bacteria. Here you can see this isoleucine in this hydrophobic patch, and we can see in these tables on the right how the substitution of isoleucine by an alanine is eliminating the antimicrobial activity of the protein and also the agglutination activity of the protein. And we have tested 3 different gram-negative bacteria species. We also studied the interaction of the protein with bacteria wall components, in particular lipopolysaccharides, which are at the outer membrane of gram-negative bacteria. And what we did is we studied a battery of different strains of E. coli, which has a truncated lipopolysaccharides at different levels. And we analyzed the activity of our protein, how this was correlated to the agglutination action of the protein. So we can see a positive correlation between the length of the lipopolysaccharide and the agglutination activity. We also characterize the interaction of the protein with lipopolysaccharides by calorimetry, also a displacement assay using a fluorescent probe. And we did also a docking analysis where we can see lipid how we can fit to our protein, and NMR titration studies that could identify some residues like this tryptophan 35, which I was talking before, which is exposed at the surface of the protein. So there is a region specific for this lipopolysaccharide binding, which is this region I highlight here in blue, from tyrosine, arginine, tryptophan, arginine, which is at the second helix of the protein. Here, which I was talking before, we can see how the interaction of the protein with lipopolysaccharides and at the same time the aggregation, protein-protein aggregation region, both are contributing to interaction of the protein to the lipopolysaccharide, and they are inducing the aggregation of the bacteria. The important thing is that the aggregation of bacteria is leading later on to bacterial death. As we can see here by staining, live rate staining, in this video we can see how the bacteria are aggregated. This is by confocal microscopy, and how bacterial agglutination is leading to bacterial death. This property of binding to lipopolysaccharide and this agglutination activity has also been correlated to high antibiofilm action. So here in this picture, we can see also by confocal microscopy how the protein can eradicate biofilms. We can see a reduction of the biofilm how we can say, the depth of the biofilm layer, and also these regions where the biofilm has been eradicated. And we can also correlate with bacterial death. We wanted to identify more precisely this antimicrobial region from our protein. And Marc Touraine, which was working initially as a PhD and later on has, with a collaboration with the University Pompeu Fabra, was doing a postdoctoral stage. He developed an algorithm which is called AMPA, which is online, where we can identify antimicrobial regions in proteins. So this was based on a library, peptide library, where from initial sequence of an antimicrobial peptide, we have substitutions by all the different standard amino acids for each position, and we can correlate the different peptides to a inhibition, minimum inhibition concentration. And with this data, he developed, he identified a propensity scale for each residue and developed this server where by assigning for each residue a propensity scale depending on the surrounding of the residue by the other amino acids and antimicrobial activity, we can identify this antimicrobial region. If we apply the server to our protein, we can identify the antimicrobial region at the N-terminus. And even if we do a screening within the RNase family, we can see conservation in this region. Some RNases, which are not antimicrobial, as RNase 5, will have less conservation at this region. Based on these results, we have tried to find out if we could work only on the antimicrobial region and design novel peptides, which could be used for therapeutics. So here in this picture, we can see how we got an initial peptide of 45 amino acid, which we know it's too big, no, for clinical assays. So we tried to do an effort to minimize the peptide length to find a good pharmacophore. We did some analysis combining the two regions, the aggregation prone region and the lipopolysaccharide binding region, even connecting with a non-peptide linker, aminohexanoid, and or alternative with a proline residue. And we could see that the peptides by NMR studies could organize, structure itself in this helix alpha, and the peptide could mimic most of the parental protein antimicrobial activity. Later on, we were having a peptide of 30 amino acid lengths, and we built a library of 500 peptides, and we did a screening of this library by substituting each position with a different amino acids. And then we test the antimicrobial activity and agglutination activity, and also hemolytic activity as a test of potential cytotoxicity in vivo. And what we found is that even if we have tested this library with more than 500 peptides, we only identified 3 peptides that could improve, slightly improve only the original peptide. So what we observe is that most of antimicrobial peptide that improve the antimicrobial activity, this was in detriment of the hemolytic activity. So when you increase the amphipaticity of the peptide, what you get in most of the cases is a higher cytotoxicity. We could conclude that probably this N-terminus peptide was already selected through evolution, has already been optimized to reduce toxicity to the host, but provide a high antimicrobial activity. We did a very preliminary and short test in vivo of our peptide, but to increase its half-life in vivo, what we did is either use D-amino acids or the substitution of arginine to ornithine as a non-natural peptide to avoid the action of proteases in vivo. So what we observe is that this peptide in an acute infection, a short infection in mice with Acinetobacter baumannii, we can see that the treatment of the peptide can improve the survival of the mice. We have a 70% of mice survival, although we don't achieve 100 survival in comparison to colistin. Colistin is a cyclic peptide that is used in the clinics as a last resource, but it's quite toxic to the kidney and the liver. So if we check the activity of the peptide, we can see that in different tissue of the mice, after 3 days, we have a drastic reduction of the infection. And at the same time, what was interesting is that we observe a reduction of the inflammatory marker, TNF. So our hypothesis is that the binding of the protein to LPS, to the lipopolysaccharides, it's blocking the LPS inflammatory action induction. So here we can see at the bottom, in the graph of the bottom, how the macrophage, when they are exposed to lipopolysaccharides, they induce this pro-inflammatory response. But when we add our peptide, we see a drastic reduction of the inflammatory response comparable to colicin. So now at the end of the talk, I'm just finishing. I would like to go back to the protein. So I have been talking of this N-terminus peptide. I have been talking of the protein action at the bacteria wall and potential internal target, but it's also important to consider the potential immunomodulatory properties. So we can say that this protein will be a multifaceted protein. So we have not only the catalytic activity, but these regions related to antibacterial activity. But I would like to finish this talk with other properties of the protein related to immunomodulation, which are also not related to the catalytic activity. So we have obtained a catalytic deployed mutant. So we have this, in the active site, we have an histidine, which is essential for catalytic action. If we we substitute this histidine by an alanine, we remove the catalytic activity, and by transcriptomic in macrophage, we could compare the pathways activated by the wild-type protein and the mutant. And we can see that there are some pathways which are unrelated to the catalytic activity. Also, we have anti-pathogen activity related and unrelated to the catalytic activity. So we see that bactericidal activity is related to is unrelated to the catalytic activity, so it's related only to the bacterial wall lysis, but the antiviral activity is related to the catalytic activity. So I want only to briefly finish the talk with a region of the protein that is interacting to tyrosine kinase receptors. And we have seen in our protein how by molecular docking, how it can feed the protein to the EGF receptor, and how MAP kinase pathway is activated by the protein and can be blocked by the presence of the antibody or against the receptor. This was previously reported by ribonuclease 5, another ribonuclease of the family, later on also reported for ribonuclease 1 within this vertebrate specific family. And here we can see that ribonuclease 3 can also interact with tyrosine kinase receptor. So we could hypothesize that this protein, which is a multifaceted protein, we could talk about a moonlight protein, a moonlight ribonuclease, because initially it was described as an enzyme, and later on other properties has been discovered. So only to finish, I want to summarize a bit the different properties that is associated to this protein. So we have this catalytic action. We have the N-terminus related to bactericidal activity. In this N-terminus, we have an aggregation, protein-protein aggregation patch. We have also a region that binds lipopolysaccharides, and here I didn't have time to talk about, but can also bind heterosaccharides like heparin mimetics, which could be useful also for some eukaryote pathogen, the binding, or can be related to potential side effects of the protein in the host. And finally, this interaction with tyrosine kinase receptor. And to finish, I would like to thank all the people from my group and the collaborators that participate in this, in some of this work from different universities, Pompeu Fabra, Birkbeck University, Borstel University, or the CSIC in Madrid. And thank you for your attention.
Thank you very much, Dr. Boa, for your very interesting presentation. Now we will have the Q&A session. We have a few questions in the Q&A box. The first question is for Dr. Papareddi. If you could please read and answer the question.
Yeah, hi. Do all proteolytic cleavage leads to HDPase formation? Actually, no and yes. So it depends on the protein. If it is if the fragments are charged, as I said, like positively charged and amphipathic, and then depending on the location, like for example, if you leave in an append tube a protein on trypsin, it will chop up everything, and it's the same with all proteases. So, and it has, once it is cleaved, and it has to bind or do its own function. But if it stays still for a long time, it will be cleaved further, and there won't be any activity. So I hope I have answered your question.
Thank you very much for your answer, Dr. Papareddi. Next, there are quite a few questions for Dr. Boa.
Yeah, well, they are quite interesting. How stable is RNase 3? Yes, in like for example, in testing serum or in the presence of bacteria in growth media, the protein usually it's about 2-4 hours. The half-life is a few hours, so it's quite stable. It's true that probably in vivo we can have different proteases, no, as Dr. Parvin was talking about, but and also we have to consider that we are working with recombinant protein, which this I go to another of the questions that has been asked. So we can say that this is a protein which is secreted to the extracellular media. So it's in comparison to other protein, it's a quite very stable protein. It has this 4-disulfide bond, so it's very resistant to in the extracellular media. But in our assays, we usually check its half-life in a few hours range time. What is the solubility rate of this protein? This protein, because it has this aggregation patch, has a tendency to aggregate depending on the conditions. So, for example, in presence of high salt, it can aggregate. But in most of the assays that we have been working, there is no problem. We can go up to more than 10 milligrams milliliter with no problem. So it's quite soluble. We produce it in, and later I go to the third and fourth question. I continue with the first question. So we produce it in bacteria, in E. coli. So this is a recombinant protein which cannot fully mimic the protein in vivo, which is glycosylated. We have done some studies, but not preliminary, and also some studies some years ago, but with only one study in 2014 in collaboration with people from the University of Uppsala, where they have purified different glycosylated forms from patients. So it's very important to be able to work on the protein expressed in a eukaryote system, and we know that it's mandatory. So this we need to do it. So always the recombinant protein in E. coli will never reproduce all the properties of the protein, but it has served us to do all the studies that I presented. And finally, the hemolytic activity. Hemolytic activity of RNase is quite low, so it's in the range. In fact, we can go to up to 200 micromolar. So this is, it's not hemolytic. It's true that the peptides, as I was showing, any change at the side chain can induce hemolysis, but the protein is not hemolytic. And the protein has not analyzed in vivo, so we cannot. Another member of his family has been analyzed in vivo, which is the human RNase-1. The protein is not cytotoxic in vivo, but we have never done any study of RNase-3 in mice, for example. Sorry about that. So I think I answered all the questions. Sorry about that.
Thank you very much, Dr. Boa, for answering the questions. Next, I would like to ask Dr. Petrova or the speakers if they have any questions.
Yeah, I can ask us one question to Esther. I was just wondering, you have done quite aggregation, something that I'm interested as well, but I was wondering, you have been working with LPS, with endotoxin. Have you tried like LTA from gram-positive bacteria, or you're more interested in gram-negative bacteria in your research?
Yeah.
The interaction
No, this is
and so on.
It's a good question. In fact, the first work we did related to binding to bacteria wall components, we analyzed the interaction with teichoic acid. And also we did some electron microscopy analysis with gram-positive species. And there is, the protein is acting on gram-positive, but we saw that the interaction with LPS was much stronger. And also because of this outer membrane from gram-negative, the results were more conclusive. So we continued. We never work back with we use gram-positive and gram-negative as controls to have always, in most of the cases in our panel, but all the further studies were done on gram-negative. But it's true that like an initial interaction with the teichoic acids, which are negative charged, there is a binding also to teichoic acid. So yeah, I agree, that's an important point also. But the agglutination action is related only to binding to LPS. So in the other case, we have interaction and then we have bacteria wall lysis, but we don't have agglutination. So it will be probably a different mechanism. So
Great. Yeah, thank you so much. Then, yeah, I don't have more questions from my side. If anyone else is interested, please.
Yeah, I have a question for Dr. Parvin, and because I thought it was really very interesting, the work. So it was very interesting, this evolutionary study, like to realize that the mice is not such a good model, for the study of these host peptides because of the difference in proteases. And so for wound healing, if you like, which you think is a good model, like for example, a good cell line model, or how can you do, which will be your best model to analyze the wound healing activity of peptides? So
I think that like it depends on what type of peptides you're using. Still, you can use mice. For example, it's easy. But for the protein like TFPI that I worked, and it is very specific. So we had to be careful which peptide works in best conditions. So we have to optimize. But of course, you can test that both in first, of course, you can start with in vitro, the cell cultures. So now we have like a lot of 3D modeling cultures are there. So multiple cells, you can just make an artificial layer. But in the end, like it's depending on what type of peptide and what is the function. But I would recommend is you should have the plasma from the same species or the same peptide. So that would be nice. But of course, yeah, you can heat inactivate if you want to use in cells. But the problem is, if it is not cells, then you should test that in the plasma.
Okay. Thank you. No, that was so. This 3D model, no, you mean no of the cell line, no, that they make a 3D, no.
Yes, exactly. So, and then of course you can also use the mice. Is very easy if you have biopsies, or maybe if you make a scar model in vitro. So you can do both.
Thank you so much. And then I think we have heard exciting talks today, and both of them were actually focusing on endogenous protein and enzymes, which can lead to antimicrobial peptides as a source of antimicrobial peptides. And I think it's very important to understand how those molecules exactly works, to how this molecule works, and it's important to understand for further development, potentially for new therapeutic strategies in infectious diseases, as it's clearly has been mentioned. It's important, and how evolution, from evolution point of view, how it's important to see that the proteins are multifunctional. They don't have just only one function. Begin with that we always are interested in one thing only, but they always can be multifunctional. Then we should always, all of us, keep in mind. And I would like to thank so much both of the speakers and everyone who is online and joining this seminar to being here today. It was very exciting to be chair of this seminar, and I'm very happy to be here today. And thank you for the invitation once again.
Thank you very much, Dr. Petrova, for being the chair of this very interesting webinar. In the end, I would like to thank once again Dr. Bois and Dr. Paparedi for their insightful presentations and answer to the questions. Thank you as well to the attendees for joining us today. I would like to let you know that the recording of the webinar will be available as soon as possible on our website, and also we can offer certificates of attendance to the ones that wish to receive them. If you can please send to us an email requesting them. Thank you very much to all of you for your time. And maybe we will see each other at the next webinar. Bye.
Bye, everyone. Thank you so much. Bye.